Hello, this video is about the Nobel Prize in Chemistry in 1907, award to Eduard Buchner for his biochemical researches and his discovery of the cell-free fermentation. You see some predecessors of his work, especially mentioned in his lecture. As you can see, you see some figures. The lecture also points to. Now we'll go to the lecture itself. Cell-free fermentation, abbreviated by me, so summarized. Buchner, that's work on the border between animate and inanimate nature of chemistry and biology. It is known since ancient times that fruit juices and sugar solutions are left to fermentate, gas develops, and fluids become cloudy, yeast deposits, and the sweet taste disappears and alcohol is formed. Lavoisier discovered that during this process sugar decomposes into carbon dioxide and ethyl alcohol. Gay-Lussac found that the weight of the sugar is almost the same as that of the product, but the yeast did remain unclear, often seen as an inferior product. In 1618, Van Heijlock saw that the yeast was spherical or elliptical, but no one believed that this was of a vegetable nature. In the 1830s, Labour, Latour, Schwamm, and Kutzing rediscovered this. Fermentation appeared as the life activity of the microorganisms. Vesalius, Liebel, and Wöhler criticized this vitalist idea. The reaction was understandable. The official product of the urea was a step to the idea that all life processes can be reduced to chemical ones. And you can understand that gave some resistance. Yeast was regarded as a decomposition product. The work of Pasteur and extent Bale made it possible to see that in nature there is no fermentation without yeast. Fermentation thus was linked with life. Swann conjectured that fermentation is the feeding of the yeast. Trabeu saw that in yeast there is a chemical body, now called enzymes, causing the fermentation. The enzymes have been observed in processes occurring in living organisms. The enzyme theory got wide support, though some plant physiologists raised objections. In the meantime, all the attempts to separate enzymes from yeast failed. 20 years of failures followed. Nageli set up a new theory about the catalytic reaction which composes sugar is totally tied to the living plasma in the yeast cells. This inspired further experiments. Cells contain plasma surrounded by the bender, but substances can travel in and out the cell. See figure 1. Boiling the cell in water, glycerine solution or sodium hydroxide for weeks was done in order to extract substances from the cell. Although these may change in the process, it was better to dissolve the membrane without impurities and chemically active solvents. Nageli and Le, Claudia Ludendorff und Vermeslin, Meyer, Fehrenbach and Amthorn achieved a certain but not complete success. 1 pot quartz with 1/5 dimetate added to yeast, then dry substances in ground and mortared. Liquid juice can be pressed out. See figure 3 and 4. In a few hours, 500 cc liquid can be contained from 1,000 grams of yeast. Hahn, assistant to Hans Buchner, was instrumental in developing the method proposing the dimetate and the press. The juice is clear yellow-brown. When heated, it separates flaked coagulated protein. Coagulable protein in the interior of microorganisms has thus proven for the first time. If the juice is diluted with water and hydrogen peroxide, oxygen formation and foaming is the result. This means a catalyse, an enzyme, is present in the organic fluid. If sugar solution is added to yeast juice, a strong fermentation of gas starches fermentation has started. The formation of carbon dioxide is accompanied by that of alcohol in the same proportions as in live yeast. In the yeast tube there are no longer any living yeast cells present. It can even be filtered, but it remains working. Delbruck and Lange put living yeast cells cannot produce any fermentation in a sugar solution. The living plasma is also not the cause of the fermentation. When antiseptic media are added to the juice, it does work. The expressed juice can be concentrated by evaporation at low temperatures. The residue is soluble in water and still works. When the residue is added to alcohol or ether, a white precipitate is obtained. This also working when solved in water. Yeast cells can be killed without the fermentation action being destroyed, for instance by drying or heating. Albert and Rapp added live yeast cells to acetone or alcohol and later ether obtained Zymen, which is a permanent yeast which cannot grow but causes a very powerful fermentation. All in all, fermentation can occur free cell, thus in the absence of living material, and this is caused by a chemical substance, an enzyme called cymase. More or less, compared to alcohol fermentation happened with urea fermentation, the conversion of urea into ammonium carbonate. Michael showed that this process is not directed by the vital activity of the bacteria, but by the agency of a separable enzyme, which is called urase. But there were experiments that caused doubt about this conclusion. Fischer in 1894 found enzymes, also using a process for rupture the cells, called maltase, lactase, and Mornelia invertase, but not a fermentation enzyme. Which is Fischer, found that when yeast is kept at ordinary temperature, it loses its activity. This was retraced by the proteolytic enzyme in the juice, endotryptase. The activity of the juice is lost even faster when digested by digestive enzymes, like pancreatin or tryptase. The presence of a proteolytic enzyme in the yeast cells was shown by Albert and Albert. It is also present in Zymen. The same is the case for live yeast seals. In recent years, research was mainly done in two directions. First, in the preparation of cymase, separation from the proteins and the rest of the enzyme in the expressed juice. This did not give any success. The separation also results in destruction of the fermentation power. And then second, follow up the chemical process in free cell fermentation. Alcohol and carbon dioxide were produced in the same rate for cell-free and live yeast fermentation. Hardy and Young showed that 20% of the sugar is built up into polysaccharides and cannot be directly reduced, but it can be hydrolyzed, and it's not glycogen. One must therefore assume the presence in the use of a synthesizing reverting enzyme. Cell-free fermentation produced glycerol and some acetic acid, but no succinate and almost no fused oils. Ellis showed that fused oils and succinate acid are not formed by decomposing sugar, but by splitting of amino acids by particular enzymes. Buchner also investigated the decomposition of sugars and found that lactic acid is an intermediary process of sugar composition before the forming of alcohol and carbon dioxide. Von Bayer, Schade, De Clos also made steps in the investigation of sugar decomposition. Their work supports that lactic acid is an intermediary product. Further research will show, or not, that alcohol and carbon dioxide are the next products. If that is the case, we know that there's a second enzyme at work. The first transforming sugar into lactic acid, the second splitting the lactic acid into alcohol and carbon dioxide. It can be assumed that both enzymes are present in the expressed juice, with the ratio depending on the state of the original yeast. Pasteur showed that no lactic acid is present in fermentation of live yeast, likely due to the fact that both enzymes are present in abundance. Meisenheimer was indispensable in this investigation of Buchner. Hahn and Young made the case even more complicated, showing that boiled juice itself is not able to cause fermentation, causing extraordinary fermentation when added to expressed juice. They also split by filtering expressed juice into two parts. Neither can by itself cause fermentation, but together they can. They assume a coenzyme essential to the activity of the zymase is unaffected by the boiling. They are also found that it can be partly replaced by phosphate. Buchner confirmed the last and found that organic derivatives of phosphoric acid can replace the coenzyme. Buchner and Klatte found that coenzymes can be destroyed by other enzymes in the expressed juice. One can see that we are still far away from understanding of the processes involved in the alcoholic fermentation of sugar and a detailed description of the zymase. Every step we make leads to new complications. The same is true for processes like chemical action of invertase, another enzyme. But we are still far away from making new enzymes ourselves. But still, a lot of progress is made in the last decades. The difference between the vitalist view and the enzyme theory is reconciled. Good elements of both are integrated. The differences between microorganisms and enzymes has become clear. The microorganisms produce the enzymes are chemical processes. Fermentations and other processes can now take place outside the organisms. The method using researching the yeast cells can be used for similar cases as Messheimer, Gaunt and Buchner did with acetic acid and lactic acid bacteria. We are seeking the cells of plants and animals more and more clearly as chemical factories, where the various products are manufactured in separate workshops. The enzymes act as overseers. We must never, therefore, let ourselves fall into the way of thinking ignorabis, we shall never know, but we must have every confidence that the day will dawn when even those processes of life we are still puzzled today will cease to be inaccessible to us natural scientists. Okay, this is the summary of the lecture. If you want to read the complete lecture, follow this link. You see some illustrations from the lecture and some information about the life and, in this case, death of the Nobel laureate. Thank you for watching. Next week we will go to the year 1908